The phage libraries can be of three main types. So first is the randomized peptide library, which consists of different peptides with fixed length. The second category is the randomized protein libraries, where we have sequence variants of a defined protein. And the third is the shotgun library, which consists of heterogeneous protein fragments. And we will discuss all these three in little detail. So when we have the randomized peptide library, we can have four different types of conformations. So, or not conformation, like the state in which they're present. So first is an unconstrained state, which has a free conformation. So the code protein can be directly linked to the peptide sequence. Then we have a constraint provision where we have a cysteine bound, for example, a cysteine bound peptide sequence joined to the code protein. This might even create some sort of a kink or a shape to the given peptide sequence. Then we have scaffolded state, where we have the peptide sequence embedded inside a protein scaffold. And the last one is the in vitro modified, where you somehow enzymatically modify the state of the peptide sequence. For example, you treat it with a tyrosine kinase and it now is attached to a P, like a phosphate group or phosphor group. The second category consists of protein variant display libraries, where we have random mutants of the same protein. So here the mutagenesis can occur by some error-prone PCR while trying to amplify it, or trying to amplify the protein variant vector inside an E. coli strain, which somehow gives the mutation to it, or you can have DNA shuffling. So the shuffling is something that happens when the mutations in different combinations occur by fragmenting the DNA, melting, and then allowing them to re-anneal. So these new combinations have even more mutations that are from the different positions than they had before the extension part of PCR. So these are all random mutants, but we can have also some site-specific mutants, such as in a particular finger of a display vector. A finger is basically something like a kink you can consider. So here you can have a sequence of NNN, where N can stand for any base, like ATGC. But in the position of the third N, for example, you can have either G or C, or you can have only U or G. So in those cases, the site-specific mutant is called NNS or NNK. And we can look at the position of the third mutant and state whether there is some sort of a mutation there or not. Finally, we have the natural peptide and protein libraries, where we display the cDNA, which can consist of, say, randomly primed cDNA fragments. So they may be of 100 to 200 base pairs in size, and they may also be full-length cDNAs. And they have the particular names. For example, if there is a randomly primed cDNA, then we have a G3P fusion, and if it's full-length cDNA, then we may have G6P fusion. These fragments can be created by some methods, like some enzymatic reaction, like after DNAs, or some physical action such as sonication or nebulization. And we can easily biopan such fragment libraries against, say, a monoclonal antibody, and see the presence of those particular libraries. Now that we have these libraries prepared with us, we can try to see the applications of such phages in our daily experiments. So we can have something like an epitope mapping, or mimetic peptides, or substrate phages, or phage antibodies. So these are four main applications of the phage libraries. If we have an epitope mapping, then we will be focusing on antibodies that can bind to certain conformational structures of the expressed phage library protein, or it can bind to some linear sequence which might be continuous, or some discontinuous portion of the epitope, which of course is dependent on the antibody epitope. So we have a provision for determining the location where these epitopes are present, or where these mutations are present, where these epitopes are present depending on the mutation frequency. We can also have peptides expressed by these phages that can mimic the antigen, a particular antigen, and then attach to those antibodies that are specific to it. We can also test the specificity of a particular enzyme by, for example, having our phage attached to a particular substrate and then sending a protease which might be able to either digest or not digest the given substrate. And if it is able to, then we are going to say that the phage is protease sensitive. If it is not, then we can say that the protease resistant phage exists. Lastly, we can also engineer antibodies from these given phages that can help to express the protein like a VH or VL segment, which is of the variable heavy chain and the variable light chain of an antibody structure. And this can be expressed outside the phage code proteins. The last topic that we can cover for these phages is called affinity maturation, which basically indicates that if you have a low affinity binding clone, you can have some sort of mutagenesis operation and then a selection criteria by which you would generate a high affinity binding clone. So you can generate the additional variability, that is the mutation, through events like a CDR walk. CDR walk is the coding region having some sort of a randomization or like consecutive mutation happening to it. Or you can have a chain shuffling, which basically means that you recombine sequences within your DNA. You can also have mutations during the PCR process, or you can have the mutator E. coli, the system, the cell machinery that helps you to replicate your cells, your DNA, single-stranded DNA, can also cause some sort of mutations to it. And this can help us to have a high affinity binding clone at the end of our entire replication process.